Alright, next topic is ATP-driven pumps. There are 4 classes of ATP-driven pumps. There's the P-type pump, the ABC transporter, the V-type proton pump, the F-type ATP synthase. The P-type pumps are called P-type because they phosphorylate themselves during the pumping cycle. This class includes many of the ion pumps that are responsible for setting up and maintaining gradients of sodium, potassium, calcium across the cell membrane. So we have an example. Calcium ATPase, aka calcium pump on the endoplasmic reticulum, or in the case of muscle cells, the sacroplasmic reticulum. So the sacroplasmic reticulum is just what the endoplasmic reticulum is called in muscle cells, myocytes. One of the functions of the ER and the SR is calcium storage. An increase in calcium concentration in any eukaryotic cell cytosol leads to specific responses by cells. The type of response is dependent on the cell type. Therefore, the concentration of calcium in the cytosol must be tightly regulated, kept very low, until a cell receives the proper signal to initiate calcium-dependent responses. The calcium ATPase pump is constantly active and is moving calcium from the cytosol into the ER or SR in the case of muscle cells. Sodium-driven antiporters or calcium-sodium exchangers on the cell membrane are also constantly moving calcium from the cytosol into the extracellular space. If we think about this and just draw it out, I have one membrane, I have another. This is my cytosol here. This is the lumen of the ER or SR, depending on if it's a muscle cell or not. And of course we have the extracellular. Now remember that calcium concentrations are very low in the cytosol. If we go back to that table, I believe it's 10 to the minus 4 millimolarity. So we have one pump, this calcium pump, that is, or calcium ATPase, which is moving calcium in this direction. Calcium 2 plus, right? It's moving calcium 2 plus in this direction because calcium concentration is much lower here. Calcium concentration compared to the cytosolic concentration is much higher here. And again, compared to the cytosolic concentration, extracellular concentration of calcium is much higher. It's a 4 log difference. This pump is also using an ATP because it's active transport. You're moving something against its gradient. So this is the calcium ATPase or calcium pump. Now on the cell membrane, you have another transporter, a cotransporter. And what this cotransporter is doing is bringing sodium in and using that to take calcium out. Okay? So the sodium concentrations are higher on the extracellular side compared to the cytosolic side. And of course, sodium is positively charged. The interior side of the cell membrane is negatively charged compared to the exterior side of the cell membrane. So sodium would like to be on the intracellular side, so its potential gradient is pointing in also. And overall, its electrochemical gradient is pointing strongly in. So sodium, the negative delta G from moving sodium with its electrochemical gradient into the cytosol is used to pump calcium out of the cytosol. So these two mechanisms are always active, and they're constantly moving calcium out to maintain that low calcium concentration within the cytosol of the cell. Because anytime calcium concentrations go up within the cytosol of a cell or portion of the cytosol of a cell, what will happen is the cell will have a specific response, and that response is dependent on the cell type. Now, how does a calcium ATPase or pump work? Well, the whole idea between these P-type pumps is that they phosphorylate themselves, right? So what we have here, you can see that you have over here, you have calcium going into the binding pocket, and ATP is bound right here, right? And once calcium gets in and ATP is bound, you sort of go to this kind of like an occluded state right here. And then here you see that ATP has been hydrolyzed to ADP, and you have the protein phosphorylated. And now the ADP is exchanged for ATP. Again, every step is unique. Okay, so here now you have ATP bound, but what's the difference between this and this is that ATP is bound in both, but over here, the protein is phosphorylated. So now what you're going to have is this leads to the opening to the other side and the release of calcium. And until calcium is released and two H pluses go in to occupy the binding pocket, you cannot go to this step. But once that happens, you go to that step, and now it's sort of back to that occluded state with ATP bound, and it's phosphorylated. And the next step is that it gets dephosphorylated, right? And once it's dephosphorylated, well, you're back to the original step, right? Because at this point now it's going to open up, and H plus will be released, and the binding site is open again for calcium. Now, the difference here between this and what we saw previously with co-transporters and just a regular transporter is that the process is moving in one direction, right? This is a single arrow. So the process will move in a single direction until it gets back to the beginning. Okay? So it's not probabilistic necessarily. I mean, there's probabilities. That's probabilities in like what calcium binding to the binding site, ATP binding to the protein, and etc. But in this case, unlike the other one, which side it's going to open to, whether it's going to go to the occluded state, and etc., that is not going to be left too much to chance, because it will go in a single direction. Note that the pumping cycle moves in one direction. Also note that each step is different than the other steps. Now, another example of a pump, a very famous pump, is the sodium-potassium pump. Now, sodium's electrochemical gradient is pointing directly in, being shown here. And this pump, what it does, it uses ATP to move 3 sodium ions out while bringing 2 potassium ions in. In both cases, both potassium and sodium are being moved against their electrochemical gradient, especially if the cell is at resting potential, meaning that you're positively charged on the outside and negatively charged on the inside. So in both cases, both sodium and potassium are being moved against their electrochemical gradient. The electrochemical gradient that sodium is being moved against is actually much steeper than potassium, but still, same thing. They're both being moved against their gradient. The sodium-potassium pump resides on the cell membrane. It moves 2 potassiums into the cytosol from the extracellular environment while moving 3 sodiums from the cytosol into the extracellular environment against its gradient. The activity of this pump is responsible for maintaining the sodium and potassium concentration gradients. This is very important, right? We're getting into how this asymmetry or these gradients are created across the cell membrane. Note that the sodium-potassium pump is electrogenic since it pumps 3 plus charges out and brings 2 plus charges in. Now, this is a fact. This statement is a fact. I mean, that is what's happening, right? Now, the conclusion that you might run into or start thinking about is, oh, okay, so this is why the exterior side of the membrane is positively charged and the interior is negatively charged. We're going to get to how the membrane potential is set up, and it's not because of the sodium-potassium pump. But the sodium-potassium pump is electrogenic, meaning that it is moving 3 plus charges out and moving 2 plus charges in. The resting potential of the membrane is dependent on many other factors, and we're going to be discussing what those factors are later. A typical animal cell devotes almost 1/3 of its energy to fueling this pump, and the pump consumes even more energy in nerve cells and in cells that are dedicated to transport process, such as those forming the kidney tubules. Where do all my calories go? Here you go. Here's 1/3 of it or more. That's where they go. Even while you're resting and not moving and doing anything, your sodium-potassium pump is working hard to maintain that sodium and potassium gradient across the cell membrane. And without it, well, there's going to be a lot of problems. if the sodium-potassium pump is inhibited. ABC transporters, structurally different than P-type ATPases, primarily pump small molecules across the cell membrane. And we have a picture of an ABC transporter here. You can see that ATP is bound, but then ATP is hydrolyzed. And that hydrolyzation right here leads to the ATP, the nucleotide, no longer binding to the protein, and that kind of leads to conformational changes of the protein, and etc. This type of ATP transporter is going to be mainly responsible for moving bigger molecules across the cell membrane against their gradient. V-type pumps are constructed from multiple subunits, polypeptide chains. The V-type protein pump transfers H+ into organelles such as lysosomes, synaptic vesicles, and plant or yeast vacuoles to acidify them. Actually, this little thing right here, you guys might want to keep track of that because that might be something that comes up later. So basically a V-type pump is kind of like a P-type pump. It's just structurally slightly different. It's using ATP to move H+ against its gradient to basically acidify some sort of organelle. Okay? Now, F-type pumps, ATP synthases, they're very similar to V-type pumps, but they work in reverse. Now, what would that mean? So, the V-type pumps, what you're doing is, here's my membrane, and you're pushing H+ in while using an ATP. ADP. And of course, eventually the inorganic phosphates will be freed. Now, a P-type pump works in reverse. What does this mean? Well, in this case, you're moving H+ against its gradient. So what we have here, you can imagine the gradient for H+ is actually pointing in this direction, okay? So this is the gradient for H+. So we're moving the gradient. So let's have the gradient for H+ be the same in this direction. So what does an F-type pump do? It will move H+ down its gradient and will take an ADP plus a PI and make ATP out of it. Right? So this condensation reaction of going from ADP to ATP is unfavorable. So it has a positive delta G, but the movement of H+ down its gradient has a negative delta G. You add those two up, the whole process is a negative delta G, and that's how you can make ATP. In oxidative phosphorylation, the last stage of cellular respiration, the electrons from NADH molecules generated during glycolysis and the citric acid cycle are handed off to the electron transport chain on the inner membrane of the mitochondria. The movement of these electrons in the electron transport chain is favorable and is coupled to the pumping of H+ out of the matrix into the intermembrane space of the mitochondria. So if you're kind of getting lost in what the intermembrane space and the matrix and of the mitochondria and all that stuff is, I will go back and look at the notes from section 1 where we covered cellular respiration, glycolysis plus the citric acid cycle, because there's a picture there that an explanation of what all these things are. Kind of take a look at it, refresh yourself, and I'll come back to this. Now, here's the electron transport chain. These three proteins are the proteins in the electron transport chain right here. So what's happening in this diagram is that the citric acid cycle is giving electrons to NADH. So basically what all this is NAD plus to NADH, okay? Now those electrons are handed off to the electron transport chain and you get your NAD plus back, okay? And now these electrons sort of move down from one protein or pass from one protein in the electron transport chain to the other, and this is a downhill movement energetically. So this is energy right here, in case it's not super clear. And then that energy, that negative delta G, is used to pump H+ out against its gradient. And what will happen is that eventually the electrons will be given to O2 and H2O is made. So this is the part where in cellular respiration where O2 is used. At the end of the electron transport chain, the electrons are captured by O2 and O2 is reduced to H2O. This is the phase in the aerobic cellular respiration where O2 is utilized. This creates an H plus gradient across the inner membrane, which is pointing into the matrix. H plus then moves down its gradient via ATP synthase, and ATP is produced from ADP and PI. So here's a few pictures just showing you ATP synthase at work, sort of like a cartoon picture. And you can see here H plus gradient being shown here. The H plus gradient is pointing in, and H plus is allowed to move down its gradient through ATP synthase. And this movement has a delta G that is negative. And then you have this thing right here, which has a delta G that is positive. And then when I add delta G plus delta G, I get a delta G for the whole thing, which is less than 0. And that's the most important thing. Otherwise, this process cannot happen. Now, this is sort of summarizing what's happening at the membrane. This is not showing you the full process, but it's just summarizing it. NADH, half an O2, plus H plus gives you NAD plus H2O. And what's coming in is ADP and plus inorganic phosphate, and you're getting an ATP. So it's basically any energy conversion process that's occurring at the membrane. What that energy conversion process is, it involves the electron transport chain and ATP synthase. So this is just saying that, okay, this is the input, hand it off to the electron transport chain, and then ATP synthase does the work, and this is going to give me ATP from ADP. Now the figure below summarizes cellular respiration in the mitochondria. And this is actually a very good picture. I like this picture very much. So we have the input from either glycolysis or fatty acids, which will enter the mitochondria straight. And then what you have is pyruvate is converted to acetyl-CoA, or fatty acids are converted to acetyl-CoA. And we saw this and we covered this in section 1. And then, of course, acetyl-CoA goes to the citric acid cycle. And if you remember, a lot of NADH is produced during the citric acid cycle. And CO2 is produced. Because CO2 is being produced within this small space of the matrix of the mitochondria, the concentration of CO2 is going to be higher in the matrix as long as the citric acid cycle is going. And CO2 is a small nonpolar molecule that can easily diffuse across membranes. And the matrix membrane, the inner membrane, and the outer membrane of the mitochondria, they're no different in composition as far as molecular-wise as the cell membrane. They're made out of phospholipids and embedded proteins and etc. So CO2 will simply diffuse out down its gradient. No need for any special transporters or anything. Now, the H plus gradient is being made here by the electron transport chain. And eventually the electrons are passed on to O2 to give me H2O. And what you have here is now H plus is allowed to go through and come back in through the ATP synthase. And then that negative delta G is used to power the making of ATP. Now, the interesting thing is that, okay, how does ADP get into the matrix and how does ATP get out? Well, there are transporters that are there for ADP and ATP. Of course, outside here in the cytosol, ATP is being used all the time to produce ADP because there's work to be done, right, to maintain life. So ADP concentration is going to be higher out here and lower in here because ADP in here is being changed to ATP. Now, ATP concentration is going to be higher in the matrix because ATP is being produced in a smaller volume, so the concentration is going to be higher. And ATP will also move down its gradient through transporters out of the matrix and out of the mitochondria. Note that O2 and CO2 can easily diffuse across the mitochondrial membranes because they are small nonpolar molecules. Okay, so we'll stop here and continue with the importance of the sodium gradient in the next video. Thank you, guys.